Hi everyone, so welcome back. So we have been discussing about the reproductive physiology, and what we have discussed so far, we discussed about the anatomy of male and female reproductive system. What we discussed about? We discussed about the anatomy of male and female reproductive system, right? Now what we'll be discussing today, we'll be discussing about gametogenesis. Gametogenesis. And what is this gametogenesis? It is the formation of gametes. What is it? It is the formation of gametes, right? In females, it is called as oogenesis. In males, it is called as spermatogenesis, right? So what we'll be discussing today, we'll be discussing about spermatogenesis. That is the formation of sperm. So about the location of spermatogenesis. So spermatogenesis takes place in seminiferous tubules of testes. Seminiferous tubules of testes, right? So now what we'll be discussing, we'll be discussing about the TS of the seminiferous tubule. You'll be really surprised to see that there are different types of cell which are present in the seminiferous tubule. So yes, so first we have this sort of long cells. This sort of long cells, and these cells are called as the Sertoli cells. Sertoli cells or nurse cells. These are called as Sertoli cells or nurse cells. What do? They provide the nourishment to the maturing sperm, right? The sperms which are undergoing maturation. We have the main sort of cells, we call it as germ cells. Germ cells, and they make a layer called as germinal epithelium. What do we call this layer? We call it as germinal epithelium, right? Now, along with that, if we go outside the section, so what we'll find? We'll find the connective tissue for sure, and we find a group of cells, right? Group of cells. These cells are called as Leydig cells. Leydig cells, right? Or we also call it as interstitial cells, will secrete the testosterone, right? So now let's just just have a look on this germ cells. So these germ cells, what happens? They divide. What do? They divide via mitosis. Sorry, they divide via mitosis, right? They divide and they increase in the number. Fine. What do? They divide and they increase in the number. Now, let's discuss about the spermatogenesis in detail. How exactly the process takes place. Sorry. Yeah, okay. Spermatogenesis, that's what we'll be discussing here. Now, spermatogenesis. Spermatogenesis has two steps. Spermatogenesis, and it has two steps. First step is the spermatocytogenesis, and another step is spermiogenesis. So what is this spermatocytogenesis? If we talk about the spermatocytogenesis, it is the formation of a spermatid. Formation of spermatid. It is the formation of spermatid, and the spermiogenesis is a process in which this spermatid, it converts into sperm or mature sperm, right? This is the maturation of the sperm, which takes in cauda epididymis. Okay, cauda epididymis, maturation of sperms, right? Now, if we talk about the spermatocytogenesis, so it starts from the germ cells, right? So this is the section we are taking here, and these are the germ cells. These are the germ cells. GC germ cell. Here they are diploid. First, what happens? It undergo mitosis, makes 2 cells, right? 2 cells. And these cells are called as spermatogonial or spermatogonial cells. Spermatogonial cells. Remember, here they are 2N. They are diploid only. Now, what happened? These spermatogonial cells, they grow in size. They grow in size, right? We'll be discussing about different phases. I'm just telling you the outline. And they are growing in size so that you'll have a clarity, deep clarity on this. Let me just erase everything, right? And let me just start with the cells only. Now, suppose this was the germ cell, become the spermatogonial cells. Now they grow in size. As they grow in size, it becomes primary spermatocyte. Primary spermatocyte. Now, this primary spermatocyte, it undergoes meiotic division, meiosis, forms. In meiosis 1, what we get? Yes, 2 cells. We call it as secondary spermatocyte, but here diploidy is N. Diploidy is N. That means they are haploid, and then they further grow into the spermatid. What? The spermatid, right? Or we also call it as tertiary spermatocyte. And hence, here the actual process of spermatocytogenesis, spermatocytogenesis ends here. Now, let's try to understand this spermatocytogenesis. You know everything what is happening. So we'll try to see it in the different phases. So there are 3 phases of spermatocytogenesis. Spermatocytogenesis. So first phase is multiplication phase. Multiplication phase. Here what is happening? This germ cells, these germ cells are multiplying to give spermatogonia. What? Spermatogonia, right? The next phase is growth phase. In this growth phase, what is happening? This spermatogonia is converted into primary spermatocyte. Primary spermatocyte. And then finally, we have the maturation phase where the meiosis is taking place and the secondary spermatocyte is being produced. Spermatocyte, and then we are getting the spermatid or tertiary spermatocyte. Tertiary spermatocyte or spermatid. Now, if we talk about the hormonal, influence. So what basically happens here is that the pituitary gland, it secretes the follicular stimulating hormone, right? What does it secrete? It secretes the follicular stimulating hormone under which the development of testes takes place, right? If it comes to the seminiferous tubule, right, it activates the germ cells to do their function, right? If it is acting on the Leydig cells, right? In Leydig cells, it is associated with the formation, or what we say, the secretion of the testosterone, right? So that's what is happening over here. I'm so sorry, the screen has shifted a bit. Yes. So now let's discuss about the I don't know why screen is moving when I'm trying to erase. Yes. Now, let's discuss about the next process that is called as spermiogenesis. Spermiogenesis. Now, this spermiogenesis is the conversion of spermatid, which is like this, to spermatozoon or the mature sperm, right? Which is like this. Okay. So what are the changes that is taking place in the conversion of this round structure to a different structure. So first thing that is taking place under the spermiogenesis is, yes, the first thing that is happening here is the condensation of the genetic material. So this genetic material is being packed over here. Packing of, no, why does this appear every time? Packing or condensation of the genetic material. The condensation of genetic material. Fine. Now, only the DNA and basic proteins are being packed here, right? And the nucleus is becoming pointed here. Pointed nucleus, right? Next step is the formation of acrosome. Formation of acrosomes, right? Now, what happens? This Golgi body, the few parts of Golgi body, few parts of Golgi body, they degenerate. Only we have this structure, this sort of structure, cristae, right? They form the acrosome. Yes. The tip of sperm, that is acrosome. And what happens here? Here we have certain proteins called as lysine proteins. Lysine proteins. We have enzyme called as hyaluronidase. Hyaluronidase. Sorry, hyaluronidase. I have written the wrong spelling. Hyaluronidase. What does it do? It secretes hyaluronic acid. What exactly it do? It helps in the thickening of the ovum, layer of ovum that we'll be discussing later, right? It thickens the ovum so that it can block polysperm. Fine. Now it is covered with the plasma membrane. The sperm get covered with the plasma membrane, right? It get covered with the plasma membrane. Along with that, cytoplasm get constricted to the tail region only, right? Okay, and that's what, oh, what is happening? So that's what we discussed about the head part. Now we have the middle piece, right? So the mitochondria, whatever is present in the cell, that becomes the spiral mitochondria like this, because mitochondria is essential for the energy production. It is essential for the energy production. Fine. So mitochondria becomes spiral here. And then in tail, we get the flagellum, right, which has cytoplasm. Okay. Now, there is one more process. So whatever is happening, right, it is happening inside the testes, right? And these sperms, right, they are attached to the Sertoli cells, right? So they get detached from the Sertoli cells, right? And then the spermiogenesis takes place. Sertoli cell, it provides nutrition to developing sperm. Okay. So that's what is happening so far. Now we have our mature sperm, right? So let me just draw the structure of mature sperm. So this is the nucleus, this is the acrosome, and this is the plasma membrane, which surround this, as well as the spiral region, right? Sorry, I'm not making good diagrams. And this is the tail region. Nucleus, acrosome, PM, plasma membrane. Middle piece contain mitochondria, that is two spiral mitochondria, and then we have the tail region. Right? So that's what is the structure of a mature sperm. I think everyone is well aware. Sorry. I think everyone is well aware about the structure of the mature sperm, if I'm not wrong. Okay, fine. Now, moving on to the spermiation, there is one important, important thing, that is the spermiation. It is the release of the sperm from the Sertoli cell. Okay, sperm from the Sertoli cells. Coming on to the viability. I told, if I'm not wrong, I have started with the viable sperm, right? That what does this reproduction depend upon? So this reproduction depends upon the the viable gametes. We should have the viable gametes as well, right? So it is the viability is a time period in which sperm is capable for the fertilization. Okay, it is capable of fertilization. Capable of fertilization. That is called as viability or the viable sperm. If the sperm they are capable of the fertilization. Okay, few things that we have to remember here is the first thing that the centrioles, right? We have centrioles as well in the middle piece. What does this have? We have centriole in the middle piece, and the arrangement of centriole is 9 plus 0. And for the axial filaments, I'll make the diagram again. Axial filaments, so you have 9 plus 2 arrangement, right? So we're just discussing about the small things, and then I'll be making a detailed structure of the sperm, right? So let's discuss about now the detailed structure of sperm. So we'll see that in the detailed structure of the sperm, what do we have? We have here the nucleus, pointed nucleus, right? This is the pointed nucleus. Here lying the centriole. And this centriole is called as proximal centriole. Proximal centriole, right? Now we have the spiral mitochondria. I'm not drawing much. Then finally we have the one more centriole, and this centriole is called as distal centriole. This area is spiral mitochondria, and we'll find few lines here, right? And this line is called as the axial filament, line of filament called as axial filament. These are spiral mitochondria, spiral MT. This is nucleus. Now we'll find in the tip we have this acrosome. This is acrosome. Now acrosome would have certain fluid here, right? And this fluid is called as glia. Okay, and then we have the plasma membrane. Then we have the plasma membrane, right? So this is the head portion. This is the middle piece. Or middle piece have a very good name, and that is the manchet. That is manchet, right? And then we have the tail, which is flagellated. Tail is flagellated. Fine. So that's what is the structure, right? So you can definitely draw the outline as well. So whatever was required, I have drawn that. What was required, I have drawn that. Okay, so this is the structure of a mature sperm. It is the structure of the mature sperm, right? So that's what is happening in gametogenesis. Or this is the spermatogenesis in humans, right? So now I think this is quite sufficient enough. It is a lot of information. Okay, so in the next video, we'll be discussing about oogenesis. Meanwhile, you can practice this. Practice the structure of sperm, the phases, different phases, growth phase, this and that, growth, multiplication, multiplication, growth, and maturation. Okay, so that is very, very important. So we'll meet you in the next video. Till then, take care. Bye-bye. Thank you for joining, everyone. Have a great day.